All right, so we are on to unexpectedly, it's part three, but I don't want to post hour-long videos, and I don't think you want to watch an hour-long video. So we are on to our last macromolecule, nucleic acids. All right, and so when you think of nucleic acids, you probably think of DNA, deoxyribonucleic acid. You also maybe think of RNA. That's also nucleic acid. And then we're going to talk about ATP, which is the energy currency of the cell. And that is also a nucleic acid. And that would also be that money molecule. And we'll talk more about that when we get to our ATP slide. So let's first talk about the brick. What is the brick for nucleic acids? So the brick is nucleotides. Okay, so each nucleotide has three components. Okay, so let's go through those components. And we're also going to talk about directionality again. So the first thing we're going to talk about is the nitrogenous base. Okay, and so as you can see, we have a nitrogenous base that is then bonded to the first carbon of a pentose sugar. Okay, and so then we go back to how we number the carbons in a sugar. So we have carbon one, and I know I'm not going to go this way because I'm going to bump into an oxygen. So then I go carbon 1, 2, 3, 4, and I look for that little chimney at the top, and that's carbon 5. Okay, so we've just went through the second component of a nucleotide, which is a pentose sugar. When you hear the word pent, that means 5. So it's a 5 carbon sugar. And on that fifth carbon, we have a phosphate group. Okay, and that functional group should look familiar to you. All right, so we see that it is the phosphorus, it gets double bonded to one oxygen, and then it's really saturated with oxygens all around it. Okay, so now let's talk about this concept of directionality. Okay, because it's going to play a big part when we look at the structure of DNA and also the replication of DNA. Okay, so what I want you to see here is on this pentose sugar. So I already identified that first carbon, that primary carbon, and it is always going to be bonded to the nitrogenous base. And that fifth carbon is always going to be bonded to the phosphate group. Okay, so I want you to kind of think when you see the roof of the house, okay, that's going to be the little chimney going up is going to be where our phosphate group is, and the 1 is going to be where our nitrogenous base is. Okay, and so now I want you to just think about these two carbons, the third and the fifth carbon, because these are going to play a really key part. Okay, so if I had an eyeball and I put my eyeball right here, okay, and I was looking up at the edge of this nucleotide, okay, I would be looking up at the 3' end, okay, because the 3' end is at the bottom, okay, and if I were up here, if my eyeball was over here and I was looking down, I'd be looking down at the 5', right? So this would be the 5', and remember, with our amino acids, we have the N terminus, C terminus, right, with our amino acids. So with here, we're going to recognize our 3' and our 5' ends, okay? And so this will basically, if you see the 3 and 5' end, that's the exterior of the DNA molecule, whereas if we were to look at the DNA double helix, we wouldn't be able to see the internal carbons because we're looking at the outside. So that's why we're always focusing on the 3' and the 5' carbons. Okay, so let's first see if there's anything we want to add to our notes. So for our general polymers they are made out of nucleotides. So nucleic acids, the monomers are nucleotides. Okay, what is the role of DNA? DNA stores our genetic code. And then we're going to pause right there. Okay, so let's look at our differences between the deoxyribose sugar versus a ribose sugar. Okay, so you can see that the difference is right here on this carbon. We're going to see a difference in what is bonded. Okay, so let's think which carbon is it? We have carbon 1, and I know this is carbon 2 because I can't go up this direction because I'm going to bump into an oxygen. So we see variation on the second carbon. Okay, and so in your notes, when you're starting to see how they differ, so they differ in the type of sugar. DNA is deoxyribose, and RNA has ribose sugar. But if we were to go through our carbons, they still are five carbon with our little chimney, carbon 1, 2, 3, 4, and up in my chimney is carbon 5. I can do the same thing with my deoxyribose, carbon 1, 2, 3, 4, and my 5th carbon up in the chimney. And the only way that they're different is on that second carbon. We either have an H if it's deoxyribose or it's an OH if it's ribose. All right? Okay. Oh, and I jumped one for you in your general section. So number 3 under general for nucleic acids, RNA allows for translation of the genetic code of DNA into the amino acid sequence of proteins. And then there's 3 types of RNA we're going to talk about: mRNA, tRNA, and rRNA. Okay. For directionality, for number 4, I'm going to show it to you in more detail, but we're going to see where we add new nucleotides. Okay. All right. So let's talk about those nitrogenous bases. Okay. So there are 5 nitrogenous bases, and there's 4 that will always be related to DNA, and there's 4 that will always be related to RNA. Okay. So the one that you won't see in RNA that will be replaced with a uracil. So you can see, well, let's rewind. There's 2 categories here. We have pyrimidines and purines. Okay, pyrimidines and purines. Now you can see there's a definite difference in their structure, right? In a pyrimidine, it's one carbon ring, okay, versus our purines have 2 carbon rings. And so a good way to remember this would be that poor primitive pyrimidines only have 1 ring. So poor primitive pyrimidines only have 1 ring. Okay, and then you can also think of our purines. So if you look at the periodic table, A and G, so adenine and guanine are purines. Ag is silver on the periodic table, so you could think pure silver, pure Ag. That would be a purine. So the difference in our purines is that we see 2 carbon rings. All right. So if you took my class, the mnemonic device I taught you in terms of how the nitrogenous bases bond together, adenine always bonds with thymine, adenine with thymine, apple in the tree. Okay, so a purine is always going to be bonding with a pyrimidine. Okay, and then what is the other one that goes together? We have cytosine that always bonds with guanine. Okay, so the only difference is that in RNA, we don't have in RNA, there's no thymine. Instead, it's uracil. So RNA always carries uracil versus DNA will always carry the thymine as its pyrimidine. Okay, so you'll just need to memorize that. Okay, so let's go into our notes and make sure we've got what we need. So for bases for DNA, it's adenine, thymine, guanine, and cytosine. And for RNA, instead of thymine, there is, fill it in, uracil. Okay? All right. So now let's talk about how the 2 nitrogenous bases bond together to start to build our double helix. All right. So let's just check. When I present this nucleotide to you and you see that nitrogenous base right here, cytosine, is it single ringed or is it double ringed? Hopefully you're saying it is single ringed and that it is a pyrimidine, okay? Because it's poor primitive pyrimidines only have one ring, okay? But if I had a guanine, okay, then I would see two rings and it would not be a pyrimidine. It would be a, check it, purine. All right. So, what is the function of DNA? Well, it stores your genetic code, and those are your genes, right? And your genes make up your chromosome, and that's these really tightly wound up strands of DNA that we can pack into each and every one of the cells of our body. And we have this genetic or instructional code for how to continue to support you being alive, right? Or for any living thing. I want to make sure I'm sticking with you on your notes here. And I think I am where I need to be. So let's just do a quick summary of the differences then between our DNA versus our RNA, okay? The sugars are different, okay? The bases are the same except for that our pyrimidine changes, right? Instead of thymine, which we see in DNA, we have uracil. In terms of strands, DNA is double-stranded and pairs at the bases, those nitrogenous bases, versus RNA is single-stranded, okay? So because it's single-stranded, is RNA going to have the double helix? No. Is DNA? Yes. All right, so now here are three different ways that we could look at the DNA molecule, and so we can start to see how those nitrogenous bases are interacting with each other. So we have our thymine, which is a pyrimidine, bonded with adenine over here. And you can see how many, well, first of all, when we see these dotted lines, hopefully we're remembering that those are what kinds of bonds? I think I heard you say hydrogen bonds. When I do that, pause and wait. I feel like Steve on Blue's Clues, by the way. I don't know if you guys even watch Blue's Clues. I feel like I just wish that we were in person. All right. So between our thymine and our adenine, we have two hydrogen bonds, okay? But you can see that the cytosine and guanines, they have three hydrogen bonds, okay? So that's just we can see on our sides here, we have our phosphate backbone and our deoxyribose sugar, which is then bonding to our nitrogenous base. Okay. And we're about to talk about the 5' versus 3' end, but I want to use a different slide for that. But we can see this is sort of the one-dimensional view of the nucleotides bonding together to build the DNA molecule. This is the double helix version, right? So now we're at the twisted ladder. And then this is just another way we could represent those nucleotides as they bond together, those monomers of DNA bonding together to collectively make the polymer that is DNA. Okay, or RNA, which is right here for us. Okay, so let's take a look at what is the key feature that's jumping out for this RNA molecule. Hopefully it is the uracil, and because it is single ringed, is it a pyrimidine? Oh crap, is it a pyrimidine or is it a purine? Hopefully you are saying it is a poor primitive pyrimidine with a single ring, but we look at this guanine right here and hopefully you're seeing that it's a purine because it's double ringed, okay? And it's bonded to our pentose sugar, which we know if we go back a few slides, that sugar, the ribose sugar, looks very, very similar to a deoxyribose sugar, except for at that second carbon, there's a slight variation, right? And then it is bonded to a phosphate group, okay? So we have one nucleotide bonded to another nucleotide, bonded to another nucleotide, and the sequence of bases is, in this case, GUAC. All right. So let's use this slide to return to your notes. Okay, in terms of the chains between DNA versus RNA, we know that the chains in DNA are two chains held together by these hydrogen bonds, okay? Versus RNA, the fill-in is single-stranded. So there's not going to be any hydrogen bonds because it's a single strand, okay? So now let's move into the section about complementary base pairing, all right? So complementary base pairing is where two strands of nucleic acids are held together by, I'm repeating myself again, but hopefully you're able to fill in the words for me. They're bonded together by hydrogen bonds, and we can see these hydrogen bonds right here. If it's a cytosine and guanine, we know it's 3 hydrogen bonds. If it's a thymine bonding to an adenine, it's just 2, and you can see that visually demonstrated for you right here. Okay, so that's your fill-in, hydrogen bonds. So a purine is always bonding with a pyrimidine, right? Purine has how many rings? Hopefully you're saying 2. Pyrimidine has how many rings? Hopefully you're saying 1. All right? Okay, and I even told you the number of hydrogen bonds in here, right? So adenine is 2 hydrogen bonds with thymine, guanine, 3 hydrogen bonds with cytosine. Okay? So now we can talk more about directionality. So in your notes in the general section, it says directionality, 3' versus 5', new nucleotides added to the 3' end of a chain. So let's talk about what the heck that means. Okay. So just to make sure we see what we're looking at here, we see the two sides of our DNA molecule, okay? So this side on the left here, the pyrimidine side, that would be one chain of nucleotides, okay? And then on the right-hand side here, this would be another chain of nucleotides. They are bonded together at the nitrogenous bases with hydrogen bonds. And so now let's sort of break down the parts of the nucleotide that we see, okay? So we know that each nucleotide has that nitrogenous base and it has a sugar and a phosphate group. And so where do those connect? So we know that the nitrogenous base, it always bonds with our first carbon, okay? And then I wrap around carbon 2, 3, 4, 5. And what's on that 5th carbon? My phosphate group, right at the top of the chimney, okay? Then I can do the same thing here with my thymine. So my nitrogenous base, my thymine, is bonded to carbon 1. Then I keep going around because I can't go this way or I bump into an oxygen. Carbon 1, 2, 3, 4. And on that 5th carbon, what do I see? I see that phosphate functional group. Okay, so you can see that they start to bond at the 3' end. So let's talk about what that means because they are going to start to stack on top of each other. Where am I going to add new nucleotides? Am I going to add new nucleotides at the 3' end or at the 5' end? And then what do I always see at these places? At the 5' end, I'm always going to see my phosphate group. And on the 3' end, I'm always going to see my nitrogenous base. Okay? And so where do we add a new nucleotide? So when we look at the outside of these, before I talk about how we add on new nucleotides, you can see that the outside of the DNA molecule is recurring phosphate sugar, phosphate sugar, phosphate sugar. And we can see that the same on this side. Sugar, phosphate, okay? So new nucleotides are always going to be added onto the 3' end. And so let's talk about what this 3' versus 5' end will be and how we see it kind of opposite on the other side. So let's go back to counting our carbons, right? So, and go back to when I referenced where I put my eyeball, right? So carbon 1, 2, 3, and we can see that is our 3' end versus our phosphate group up here is added to our 1, 2, 3, 4, 5th carbon, so the 5' end, okay? But if we look at this side of the DNA strand, when we look across at the houses across the street, they all look upside down, right? So I want you to kind of think of it as a road, right? One is going this way, so from 3' to 5' is going this way versus the other side of the street, 3' to 5' is going in the opposite direction. Okay, but it's still following the same rule. So let's follow it. Let's look at our sugar here. Okay, so I have my first carbon, carbon 1. I know I'm not going to go this way because I bump into an oxygen. So I have carbon 1, 2, 3. There we go. That's my 3' end. Okay, these sugars are bonded to a phosphate, right? The sugar phosphate backbone. And then we have our next sugar, which we again will see our phosphate group will be attached to the 5' end. But let's just check by counting. I have carbon 1 here, which we all know is attached to our nitrogenous base. In this case, it's our adenine. Okay, and am I going to go this direction towards the oxygen? Nope, I'm going to count the other way. Carbon 1, 2, 3, 4, and on my 5th carbon, what do I see? I see my phosphate group. So from 3 to 5 on one direction, and 3 to 5 on the other direction. And this is how we see the directionality of adding nucleotides to our DNA molecule. Okay, so something to keep in mind, new bases are only ever added at the 3' end. Okay, and we're going to get more into that as we get deeper into DNA synthesis, but we only are going to add new nucleotides at the 3' end. All right. So now let's talk about ATP, which is our last. That's our last form of nucleic acid that we're going to talk about. And so what does ATP stand for? And it's waiting to be filled in your notes. ATP stands for adenosine. You see it right here, adenosine triphosphate. And so let's kind of break down a lot of the familiar chunks within those words. When I hear adenosine, I'm immediately thinking of the nitrogenous base, adenine. And is adenine, I look at it, there's that adenine. Is that a purine or is that a pyrimidine? I'm going to give you some think time and hopefully you are saying it's a purine. Why is it a purine? Because it has, that's right, 2 carbon rings. Okay, so we have our adenine, which is bonded to a pentose sugar, we can see, and now the triphosphate part of adenosine triphosphate. Why is it called triphosphate? Because count them, 1, 2, 3 atoms of phosphorus. All right, so adenosine triphosphate, that's our money molecule. This is like the energy fuel for all cellular reactions, everything going on in the body. It's fueled by ATP. And why? Why is that? Well, because when you snap off one of these phosphates, okay, when I break off a phosphorus from this triphosphate, it releases a tremendous amount of energy. You get 7.3 kilocalories of energy every time you break off one of those phosphates. And hopefully you're thinking, when you break off a phosphate, how do we do this? Well, let's look. Hopefully you're immediately thinking back to that process whereby we wedge in a water molecule and we are able to break bonds. Hydrolysis. Hydro, water, lysis, meaning to cut, cutting those bonds by wedging in water. So let's look at this. When we add water to ATP and it undergoes a hydrolysis reaction, so we snap off one of those phosphates, we release energy, right? And that's what we're looking for. We break bonds And in this case, when we break off that phosphate, we release a bunch of energy because there's just so much locked in between these phosphate bonds. Okay. And so when we do break off that phosphate, it becomes ADP, or adenosine diphosphate, di meaning two. Okay. So let's see what we need to add to our notes. So number one I gave to you. So it is a ribose pentose sugar plus an adenine base. Okay. And then what's the triphosphate part? That means it's 3 phosphate groups. And then what is the function of ATP? Function, it's a high energy molecule because the last 2 phosphate bonds release energy when broken. They release energy when broken. Okay. And so this is why we eat food. Ultimately, this is the reason that you're eating food. You're eating food not only because it tastes good, but because you are taking in the food to ultimately add a phosphate group onto ADP, okay? So that you can break that phosphate group off again and use that energy, okay? So when you spend money, you got to make money. So when you spend those phosphates by, let's say you go for a run, you got to eat more food to replenish that store to get more phosphates added onto your adenosine diphosphates. Okay, so what is ATP used for? It's used for synthetic reactions, muscle contractions, like when you're working out, the transmission of nerve impulses, so your nerve cells communicating to each other, that uses energy, requires ATP. Okay, so now let's wrap it up. You have gone through all of the macromolecules with me. This is me giving you a golf clap. I could also cheer, but I'm in my apartment, so it would feel a little weird to cheer in my living room. So let's go through our summary chart. So we have now covered the 4 macromolecules. So in summary, carbohydrates. The monomer are monosaccharides like glucose, fructose, or galactose. Okay, I had those key disaccharides that I wanted you to remember. Maltose is glucose and a glucose. Sucrose is glucose plus a fructose. And lactose is a glucose plus a galactose. Okay, how do I bond 2 monomers together? You're shouting into your computer at me, dehydration synthesis. Okay. And when I bond a whole bunch of monosaccharides together, I get the carbohydrate polymer form, which is a polysaccharide. Okay. And so we learned about starch, which is plants' energy storage, or glycogen, which is animal energy storage. And we talked about the relationship between glycogen and excess fat in your body, right? If you ate enough carbohydrates that your glycogen stores are all filled up, you then put in extra glucose into your body by, let's say, eating a cupcake, right? Your body's going to immediately convert that extra glucose to fat because your glycogen stores are already filled. Cellulose, we learned that's the most abundant molecule on the planet. Why? Because it is the foundation structural carbohydrate for building plant cell walls. And then we learned about chitin. Chitin builds the cell walls of fungus, and it also builds it for organisms like arthropods or other exoskeleton organisms. Okay, so then we move on to lipids. We learned the 2 big categories of lipids are fats and steroids. Steroids are, you can identify them via their carbon rings. Fats are fatty acid chains, okay, that are bonded to a glycerol. All right. And you can have either saturated fats or unsaturated fats. Saturated fats are solid at room temperature because they just stack right on top of each other because they're so saturated with hydrogen, versus our kinky fats. And by kinky, I mean kinky like this. And they are unsaturated because they are not filled with hydrogens. Instead, there are double carbon bonds that make them look kind of kinky. And because they're kinky, they are liquid at room temperature. They do not just stack nice and easily on top of each other. So our polymers for lipids, we learned about fats and oils, critical for energy storage, phospholipids, where remember we removed one of those fatty acid chains, we replaced it with a phosphate group that was polar in nature, right? So it's hydrophilic, and then we had those hydrophobic fatty acid chains, and those are going to be really critical for building cell membranes. We learned about steroids and waxes, okay? And then we are moving on to proteins. So proteins, I want you to remember that the monomer is amino acids, and that this setup for the molecule is always going to be the same. You're always going to have your carboxyl group, but remember, it's going to have a negative charge when you lose this hydrogen right here, and it transfers over to the amino group, right? And the amino group becomes overall positive. The carboxyl group becomes overall negative. We have our hydrogen that hangs out upstairs, and then the R group on our amino acid, which dictates which type of amino acid it is, right? So, and those R groups could be extremely complex, like tryptophan, all the way to very, very simple glycine, which was literally just one hydrogen, okay? But whatever that R group is, it will dictate the behavior of that amino acid, okay? And when I, through dehydration synthesis, bond a whole bunch of amino acids together. And hopefully you're thinking, oh yeah, those bonds between amino acids, what are they called? You're shouting at me, peptide bonds. So when I have a whole bunch of amino acids bonded together via peptide bonds, I start to get that primary structure, which is the sequence of different amino acids in my chain that can then fold into our secondary structure, which is all based on the charges on the carboxyl and amino groups. Then I can add in that tertiary, and in that primary structure, I have two options. What are they? Alpha helix or beta pleated sheets. Then I have that tertiary structure, which is based on the R group, right? And hydrophobic R groups are going to want to hang out with hydrophobic R groups in that long chain, and the hydrophilic are going to want to hang out with the hydrophilic. And then we add in quaternary structure. Another polypeptide chain comes in to interact with that polypeptide chain. What are the polymers? We see them over here. All of those really critical proteins in the body. Okay, then finally, this video was all about nucleic acids. So the monomer is nucleotides, and we learned those three critical components of a nucleotide. The phosphate group, our pentose sugar, which will either be, depending on if it's deoxyribonucleic acid or ribonucleic acid, the sugar will either be ribose or deoxyribose sugar. Okay, and then we have our nitrogenous base, which is always at which carbon? Hopefully you're saying it's the first carbon, right? So bonded at the first carbon, whereas our phosphate group is always bonded at the fifth carbon, right? And then what are those nitrogenous bases that we have? For DNA, it's always going to be adenine, thymine, cytosine, and guanine. And for RNA, it's going to be exactly the same, except you're replacing that thymine with uracil. All right. And so what are the polymers of nucleic acid? We went through DNA, our genetic code, the genetic code of all living things, RNA, which is critical to building our DNA molecules, and finally ATP, that money molecule that is critical for all cellular energetics and all the cellular processes that we carry out in our body. Okay. So you guys are so amazing. You're so smart. And I know that was a ton of information, but now is the time for you to really go back through your Unit 1 hyperdoc. And I would say go through this important molecule summary chart and start to self-assess. Where have I mastered material? Where is my understanding lacking? And I need to go back and do some more studying. Perfect example would be if you cannot fill out this molecule summary chart without looking at my example, my template, that means you need to be able to go back and do that on your own. This is basics that you need to know, okay? And so the other key component is like I mentioned, I want you to go back to your Unit 1 HyperDoc and start to go through those learning targets. At this point, we have covered the learning targets for Unit 1, and so now it's review time, baby. So I am really proud of you for getting through a unit with me. We're going to do some more practice applying the concepts of especially proteins and protein folding. And don't worry if you're thinking, I didn't get enough DNA information. I want more. Well, we're going to dive much more deeply into DNA synthesis in units moving forward, specifically related to heredity. Okay. So I'm really proud of you guys. You've got this. It's study time. Let's go. I'm also going to throw in a who ride because I'm just like feeling like I'm just going to who ride on out of here.